Okay, we're going to talk briefly about nerve signal transmission and reflex arcs. There are a couple of things associated with the nervous signal transmission. There is a electrical charge that propagates along the axon of the cell, and then there is a neurotransmitter that is released at the end of the axon, which signals to the adjacent cell, or whether that's a muscle or another nerve cell, that the signal needs to continue. So there is two kinds of processes that we're going to explore there. And then there are reflex arcs, and reflex arcs exist as protective mechanisms. They are things that our body does without any conscious thought that protect us from falling down, from injuring ourselves, also helping to maintain homeostasis. So let's take a look at these different aspects here. First off, you should know that the cell membrane has a charge, and that is a very small charge. It's like negative 90 millivolts, which also explains why it doesn't take much of an electrical shock to cause problems for a person. But we're looking at a resting neuron. It is polarized. That is, the inside of the cell has a certain charge. The outside has a different charge. In this case, it's a positive charge on the outside and a slightly negative charge on the inside because of a separation of some ions. Now, we talked back in the beginning of the semester about sodium-potassium pumps, and when we were talking about cell transport mechanisms. So this is going back to that. We have these sodium and potassium pumps that move sodium out of the cell, potassium into the cell, but it moves 3 sodium for every 2 potassium, which ends up causing this slight charge that occurs. Now, when this charge starts to get reversed, that is an action potential. So you can see here we've got positive on the outside, negative on the inside, but for whatever reason, we start to have an exchange of sodium and potassium at one end of the cell. And what happens is the sodium goes in, the potassium goes out, and that changes the charge in that region. Now, if you remember facilitated diffusion, that's where we have proteins that can act as gates. And so these are electrically gated channels. And when we get this reversal of charge, then this area adjacent to it, those channels open up, which cause more sodium to move in and more potassium to move out, which causes the area adjacent to that to have its channels open, and so forth, so that the signal just propagates down the length of the axon. Now, if the action potential or nerve impulse starts, it is propagated over the entire axon. It doesn't just go part way and then stop. If there are branches, it's going to continue to go through the whole thing. Now, nerve axons can either be myelinated or non-myelinated. Non-myelinated nerve signals tend to travel much slower than those that are myelinated. And we'll talk about how that comes into play here in just a second. But here is your standard nerve impulse. And what we see here is sodium going in. That changes the overall charge to a negative charge on the surface, and that causes gated channels to open elsewhere, and it propagates down the length of the axon. Now, once this has occurred, the cell has to repolarize. And so sodium and potassium pumps begin moving the sodium and potassium back to their original locations. Meanwhile, while that's happening, the signal is continuing to propagate down the axon. This results in the nerve resetting itself, basically, while that signal is moving, and it does prevent a nerve signal from going back the opposite direction. This is recharging as this is discharging all the way down. And so as it gets further down, this area up here recharges and is ready once again to send a signal. That does mean that you cannot send a signal immediately after a signal has been sent. There is something called a refractory period where we have to repolarize the nerve. Yeah, again, repolarization. Potassium ions rush out of the neuron after the sodium has moved in, which repolarizes the membrane briefly, and then the sodium-potassium pumps using ATP restores the original configuration. So again, looks something like this. Potassium moves out, we get positive back on the surface, and then the pumps continue to move it. At this stage, we still would not be able to send a signal until the sodium-potassium pumps have actually reset the potassium to being the predominant ion inside the cell and the sodium to be the predominant one outside of the cell. Now, once a signal propagates all the way down to the end of the nerve has to communicate with the next structure, whether that's a muscle or another nerve. And it does this by releasing a neurotransmitter. So these positive, negative ions open some different channels which allow the release of neurotransmitter. And the neurotransmitter is released into this little gap called a synapse, which lies between the sending cell and the receiving cell. And this is at the nerve's axon terminal. The dendrite of the next neuron has receptors that will pick up the neurotransmitter and open some gates, which will allow sodium to pass in and potassium to pass out, and we start the whole thing over again. So an action potential is started in the dendrite. So dendrites in nerves are the receiving portion, whereas axons are the sending portion. Let's take another look here. Just basically the signal at the synapse. We've got this electrical signal that has traveled down the axon, and it results in the fusion of these vessels with neurotransmitter to dump that neurotransmitter into this gap. And there you have ion channels, which are sensitive to the neurotransmitter, and when the neurotransmitter binds to it, it causes those channels to open up. Now, this area right here, this synaptic cleft, is where a lot of pharmacology takes place, because we have drugs that can slow down the reuptake of this neurotransmitter. The body doesn't waste anything that it can, that it doesn't have to, so it reuses neurotransmitter, and to do that, it has to take it back up again. So we can have selective serotonin reuptake inhibitors as an example, which slows that process down and leaves the neurotransmitter out in that cleft just a little bit longer. Here's just an image to give you a better idea of how this works. We've got a signal, again, that's gone to the axon terminal, release of neurotransmitter into the cleft. Step by step of what we've just said. So vesicles fuse with the plasma membrane, and then the neurotransmitter is released into that synaptic cleft, and then the neurotransmitter binds to the receptor on the other side. Now, Here's the neurotransmitter being received. It has bound to that protein channel, which opens up and allows the sodium to go in. And ultimately, the neurotransmitter is broken down, released, and reabsorbed by the sending cell. Once it does that, it closes the channel. So that was a short and simple explanation of how a neurotransmitter or how a nerve signal goes. This is by no means the full depth of this, but I just want you to understand the essentials. Now, a reflex arc involves a couple of different cells. It does involve a sender, a receiver. It involves a sensor that picks up some bit of information that is necessary to initiate this reflex arc. So the reflex arc is rapid, it's predictable, and it's involuntary. The reason it needs to be rapid is, again, protective. Predictable, you can initiate a reflex by just providing the appropriate stimulus. And involuntary, I cannot in any way stop my reflex actions from occurring. If you thwack my knee properly with a rubber hammer, I will kick regardless. So this occurs over reflex arcs, and a reflex arc is a direct route from sensory neuron to an interneuron, which processes the information and then sends a signal back out to an effector. So it looks something like this. Say I've got skin and I stimulate it somehow. Say I step on a nail or I get stuck with a pin. That's going to send a signal along a sensory neuron to an interneuron, which is located, in this case, in the spinal cord. And most of our reflexes are located in the spinal cord and processed through the spinal cord. So that integration system, by being in the spinal cord, it lowers the distance for a reaction to have to occur. Because if this had to go up the spinal cord to the brain, get processed in the brain, and send a signal down, that's a few extra milliseconds where additional damage could occur. Then we send out signal to usually a motor neuron, which causes an effector, in this case a muscle, to contract. So if I were to step on a nail, I would get a signal which would cause me to contract my leg muscle, which would take my foot off of the nail. So types of reflexes and regulation: activation of skeletal muscles, for example, when you move your hand away from a hot stove. Autonomic reflexes also come into play, and they are not protective like the somatic reflexes are, but they adjust things like smooth muscle regulation, which can dilate or constrict blood vessels, which will in fact regulate heart rate, blood pressure, regulation of glands. For example, we sense that it is hot. We send a signal out which causes our glands to start producing sweat. And digestive system regulation. We have sensory receptors in the stomach which become aware of stretch and start contracting the stomach muscles in response to that. Again, the most common one that everybody is really aware of is the patellar or knee jerk reflex, and it's an example of that very simple 2-neuron reflex arc. Now, this is all we're going to discuss about the nerve signals and reflex arcs. Again, there's a lot more that we could go into depth with, but this is the simplified version and is sufficient for our purposes.